In the past few chapters, we have discussed the replication of DNA, the transcription of DNA genetic information to RNA, and the modification of RNA. Now it's time to see how messenger RNA, transfer RNA, and ribosomal RNA work together in the polymerization of amino acids to build proteins. Now is a good time to review the central dogma of molecular biology. The complementarity of the two strands of DNA is essential for its function as the storehouse of genetic information, since its information must be copied or replicated for each new generation. Expression of the genetic material stored within DNA is used to direct synthesis of proteins. This process involves the transcription of DNA segments to produce a complementary strand of RNA. Messenger RNA is translated in the ribosome, which is a cellular particle consisting of protein and ribosomal RNA. At the ribosome, transfer RNA molecules, which carry amino acids, recognize sequential sets of 3 bases known as codons. The recognition occurs through base complementary pairing with messenger RNA. The ribosome then covalently links the amino acids carried by successive transfer RNAs to eventually form the protein. The correspondence between amino acids and messenger RNA codons is known as the genetic code. There are a total of 64 codons, which consist of a set of 3 nucleotides. Of the 64 codons, there are 3 that exist as a stop message. These codons are UAA, UAG, and UGA. Upon reading these on the messenger RNA, the synthesis of your protein would then terminate. The remaining 61 codons represent, with some redundancy, the 20 standard amino acids that are found in proteins. Table 4.22 shows which codons specify which amino acids. For example, phenylalanine is coded for 2 different codon sequences, either UUU or UUC. Methionine has only 1 codon, which is an AUG. You can see serine, for example, has 4 different codons. Same thing with proline, leucine, valine, alanine. So there is multiple redundancy that is occurring. There is absolutely no need to memorize this table. It will be provided for you on the exam. The front cover of your textbook also has this image, which has the coding that you can use for homework or other assignments. We're going to first look at transfer RNA. And with transfer RNA, it's not uncommon for these RNA molecules to contain multiple modified bases. This table lists some of the modified bases, but the most common ones that you would see in transfer RNA would be pseudouridine, which is represented with the psi symbol. And another common one would be dihydrouridine with the highlighted boxes showing you the differences between the normal base that you would find and the modifications that are made. Another common base that's not seen on this table would be inosine, or symbolized with an I. We've talked about inosine in previous lectures. What is the role of the transfer RNA? Well, as its name implies, it's going to transfer something, and in this case, it's going to transfer your amino acids. Therefore, the tRNA carries out the work of bringing amino acids to the ribosome for protein synthesis. A bacterial cell will contain roughly 30 to 40 different transfer RNAs, and a mammalian cell could have as many as 150 different types of transfer RNA. The structure of all transfer RNA molecules are very similar, as they must be in order to interact interchangeably with the ribosome and with protein factors required for translation. Each transfer RNA molecule contains between 60 and 95 nucleotides, with up to 25% of these nucleotides being post-transcriptionally modified. Many of the base pairs will generate a short loop and stems, which is commonly called a cloverleaf secondary pattern. You can see that with our image, we have essentially a cloverleaf pattern. It's more apparent on the two-dimensional drawing compared to the three-dimensional drawing. It is the 5' end of the tRNA that pairs with bases near the 3' end to form the amino acid acceptor stem. Here's our 5' end. Here is the 3' end. And notice we have base pairing, which forms this acceptor stem. The amino acid itself is going to attach to the 3' end of the transfer RNA. We also can see that there are other base pairing stems that occur. which result in forming several different loops. The DHU loop often contains the modified bases dihydrouridine, and the T-Psi-C loop often contains the sequence thiamine, pseudouridine, and cytosine. Sometimes there's an extra arm or an extra loop, and as the name implies, a variable loop could range from 3 to 21 nucleotides in different transfer RNAs, and there is no consistency with the nucleotide bases that are found in that variable loop. And finally, the most important loop would be the anticodon loop. This includes the 3 nucleotides that will pair with a messenger RNA codon. Therefore, this is the communication that occurs between messenger RNA and the transfer RNA, and it directs what type of amino acid should be brought in for synthesis. Now, due to the base pairing between different elements, we have these stems that are created. And a transfer RNA then folds according to secondary structure into this L-shape structure. And through these stacking interactions, the hydrogen bonding interactions, we get a stabilized structure. Looking at that three-dimensional structure, we can see that virtually all of the bases are buried in the interior of the transfer RNA, except for the anticodon region. In this anticodon region, there is a triplet of nucleotide bases that stick out and therefore can interact with the messenger RNA. The other region where we have some nucleotides sticking out would be at the 3' end. And this is where we have the CCA tail to the 3' end. And these are the 2 most important sites for translation site recognition and amino acid attachment. During translation, transfer RNA molecules will align with messenger RNA codons. And the alignment is based on base pairing in an anti-parallel fashion. Here is our messenger RNA, and it is being read in 5' to 3'. The codon is UCC, which could then base pair with the anticodon in the transfer RNA. U and A will base pair, C and G base pair, and we can have C and G base pair again, and that is the recognition to bring in that specific transfer RNA, which has an amino acid attached to it. So based on that pairing, at first glance, it would seem this sort of specific pairing would require the presence of 61 different transfer RNA molecules. One to recognize each of the codons listed on slide 3. But there is a fact that many transfer RNAs can bond to more than one of the codons that specify their amino acid. The third anticodon position and the 5' codon position experience some flexibility, or what we call wobble, in the geometry of their hydrogen bonding. This means that the first two positions on the codon have to do your conventional base pairing, but the third position in the codon will experience some variation, and we do not have to do traditional base pairing. So with this less discriminating position, we have a little bit more flexibility. In the wobble hypothesis, the first two bases of the codon pair will always come together in the standard fashion, and their recognition is precise. This means that the codons that differ in either of their first two bases must be recognized by different transfer RNAs. The codons UUA and CUA, which both code for leucine, are read by different leucine transfer RNAs. The first base of an anticodon determines whether a particular transfer RNA molecule reads 1, 2, or 3 kinds of codons. For example, C or A will only read for one codon. G or U will read for 2 different codons. Typically, G would want to base pair with C, U would base pair with A. That's why they're in bold. But according to the wobble hypothesis, G could also base pair with U, and vice versa, U could base pair with G. And then, if we have I, inosine, that could read for 3 different ones. It could read for C, A, or U. The degeneracy, or repetition, of the genetic code arises from the imprecision in the pairing of the third base of the codon with the first base of the anticodon. Let's look at a couple of examples. The anticodon for alanine transfer RNA is IGC, when we are reading from 5 prime. When we're reading it from 5 prime to 3 prime. This means the alanine transfer RNA molecule could recognize any one of 3 different codons. It could recognize GCU, GCC, or GCA. Notice the conserved nature of the first 2 bases in the codon. But since the anticodon in the transfer RNA contains inosine, it could either do a recognition with C, A, or U. That's why there are 3 different codons that will accept the alanine transfer RNA. In another example, the anticodon for a glutamyl transfer RNA is UUC. Again, reading that 5 prime to 3 prime. This means the anticodon in the transfer RNA could recognize either a GAA or a GAG in the messenger RNA codon. Again, the first 2 positions of the codon have to do your conventional hydrogen bonding. The C and the G pair, the A and the U pair. But in the 3rd codon position, that could undergo the wobble hypothesis, and since the anticodon contains a U, that U could either base pair with A or G. Therefore, the glutamyl transfer RNA of UUC is going to recognize 2 different codon sites. Aminoacylation is the attachment of an amino acid to a transfer RNA. This reaction is catalyzed by aminoacyl transfer RNA synthetase. To ensure accurate translation, the synthetase must append to the appropriate amino acid to the transfer RNA bearing the correct corresponding anticodon. Aminoacyl tRNA synthetase interact with both the transfer RNA anticodon as well as the aminoacylation site to ensure high fidelity, making sure that the correct amino acid is attached to the correct transfer RNA according to the anticodon. The first step in the attachment of the amino acid is to activate the amino acid. The amino acids react with ATP to generate aminoacyl adenylate, where the lone pair electron on the oxygen will do a nucleophilic attack on the first phospho group, which then is going to cleave pyrophosphate. That will undergo hydrolysis to give you 2 inorganic phosphate, and as we know, that releases a bunch of free energy, making this reaction irreversible. Now, the aminoacyl transfer RNA synthetase catalyzes the formation of an ester bond between the activated amino acid and a hydroxyl group of the ribose at the 3' end of a transfer RNA, which results in the aminoacyl transfer RNA. So here is the 3' end of the transfer RNA. The 3' hydroxyl group will attack the carbon, which then cleaves your bond and now leaves you with your amino group attached to the transfer RNA. The transfer RNA molecule is now said to be charged with an amino acid. It's ready to deliver that amino acid to the ribosome. Nearly all cells contain 20 different aminoacyl transfer RNA synthetase enzymes. We need 20 because that corresponds to 1 enzyme for each type of amino acid. Although all aminoacyl transfer RNA synthetases catalyze the same reaction, they do not exhibit a conserved size of quaternary structure. These differences give rise to active sites that are specific to certain amino acids. For example, threonyl transfer RNA synthetase contains a zinc ion at the active site that interacts with the hydroxyl group of threonine. Valine is a similar amino acid in the overall structure to threonine, but it lacks the hydroxyl group and thus will not join to the threonyl transfer RNA synthetase. Serine is smaller than threonine, but it could occasionally link to the threonyl transfer RNA synthetase because it does have the presence of the hydroxyl group. So there needs to be a mechanism that removes the unwanted amino acids that could possibly venture in to the active site. To accomplish this, many synthetases have editing sites. Threonyl transfer RNA synthetase has such an editing site, which is in addition to the active site. You can see here, there's an active site, and then there's also an editing site in the enzyme. The purpose of the editing site is to remove an amino acid that is inappropriately joined to the 3' transfer RNA synthetase. The mechanism that occurs is the CCA arm of the 3' transfer RNase, represented right here, will swing the amino acid portion into the editing site where the incorrect amino acid is removed through hydrolysis. So in this example, the incorrect amino acid, valine, fits into the active site, but because it does not have a hydroxyl group, it cannot interact with the zinc ion that is in the 3' transfer RNA synthetase active site. Since it lacks this interaction, the valine does not stay in the active site. Instead, the CCA arm is going to swing up into the editing site in which you will cleave the valine. Once that occurs, then the transfer RNA is removed, and a new transfer RNA will come in, and hopefully this time we'll get the correct amino acid attached. Overall, proofreading can be thought of as a double sieve. In the first case, we are going to discriminate based on sizes and maybe polarities. So the amino acids that don't have the correct size, polarity, won't even make it into the active site where the attachment to the transfer RNA can do its binding. But if you have amino acids that are going to be close enough in shape where the amino acid finds its way into the active site, it then is going to have a second check by going through the editing site. And then based on interactions in the editing site, the amino acid is either approved or denied. So that is what we call the double sieve proofing of transfer RNA. Transfer RNA is only one component of translation. We also need to have ribosomes that are going to read or attach themselves to the messenger RNA. So here's a look at a couple of different types of ribosomes. To get the proteins synthesized from the genetic information that's found on the messenger RNA, the amino acids must get together so the amino acids can be covalently linked in a specific order. That's the purpose of the ribosome. The ribosome is a large complex containing both ribosomal RNA and protein. A ribosome consists of a large and small subunit containing RNA molecules, all of which can be described by their sedimentation coefficient. That's the difference between the 50S, the 70S, and the 30S. When do they sediment? Through centrifugation. If we look at E. coli, the ribosome sediments at 70S and is composed of two subunits, a large 50s subunit and a smaller 30s subunit. The 50s subunit is composed of about 34 different proteins and 23S RNA, as well as a 5S RNA. The 30S subunit contains 21 different proteins and a molecule of 16S RNA. For many years, the protein components of ribosomes were thought to orchestrate protein synthesis, with the RNA acting only as a scaffold. This begs the question, how can complex proteins be synthesized if complex proteins are needed for protein synthesis? It's the same thing as which came first, the chicken or the egg. So this is our chicken or the egg question for biochemistry. Well, the answer is proteins don't build proteins. It is the RNA that exhibits catalytic activity. The RNA in ribosomes catalyze the synthesis of proteins, and proteins are only added later to act as a scaffold to enhance efficiency. The mapping of the folding pattern of ribosomal RNA has allowed scientists to discover the catalytic regions of ribosomes. The next lecture will look at the reaction details of protein synthesis.